Okay, so I think we can start the class, right? So in the last class what we were talking about plastid, right? So we have discussed the endomembrane system, mitochondria we have completed, and we were talking about plastid. Plastid or ki kotha monot ase, we know that this is a cell organelle that is present only in case of a plant cell. Talking about the plastids we have talked about types of plastid. How many types of plastids? 3 types, that is chloroplast, chromoplast and leucoplast. Talking about plastid, another point that you have to know is certain types of plastids could be converted from one form to the other form. If you talk about chloroplast and chromoplast, we have discussed certain examples which tells us that chloroplast ki hole convert hoye jay chromoplast hole, but it is never vice versa. Chromoplast ketiyao chloroplast hole convert hobo nware. We have given the example of ripening of a fruit, right? We have talked about a ripe tomato and a raw tomato, right? Or maybe we have talked about a ripe chili and a raw chili, right? So ebar ripen hoy joar pisot, why do we see the color? Because of the pigments, right? And once the pigments are present, green color pigment thakile tak ami ki boli kom chloroplast, and if suppose the other colored pigments are present, then it is called as chromoplast. The third was leucoplast. The characteristic of leucoplast was it is colorless and it is divided into 3 types, or it is of 3 types depending on what is the storage material. Some of the plastids contains what? Starch, right? Kobat oils or fats asile aru kobat proteins asile, right? Or starch asile. Yeah, please come in. So today what we will do is we will discuss about chloroplast in detail, since we have talked about it before also. So as a heading koriba chloroplast and draw the diagram first. What is chloroplast? The green color plastid is called as a chloroplast. Rough diagram eta aka. Please come. Camera audio to on ho aise, please mute yourself. কোনটো মই এটা যোনটো লিখি আছো হেটো মই যোনটো লিখি আছো হেটো এইটো সাইড this one thylakoid t এটো ন thylallakoid thylakoid রিফ্লেকশন পড়িছে নেকি তাতে ওকে So whenever you study a chloroplast, these are the parts that you have to remember, right? So we'll explain the parts and after that we'll start writing the notes. Is the diagram over? 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 Okay, please do it fast. Okay, do it fast. FRET, Fret canal. Fret canal, CHA. Ribosomes ho upor likhiso stroma, STROMA. So a chloroplast or structure, this is something that you have to study during our photosynthesis class also. Right. So it's important that you know the structure, certain terms, certain words that are related with photosynthesis also, because we know if a plant has to conduct photosynthesis, it has to undergo light reaction. Light reaction hobo lagile, it has to have pigments, and pigments would be present obviously in the chloroplast. So chloroplast or code pigments thake ki thake, what are the terms, what are the structure, or what is the structure of the chloroplast, ami yate will study. So we are not going to draw the structure again in photosynthesis, right? So okay, I think it's over now. Right? Okay, talking stroma lamella or fret canal, FRET, fret canal, CHANNEL. Channel kwa canal kwa. Oh, hey pine likhise grana, GRANNA. Okay. Okay, so what we know, chloroplast is a double membrane structure. If you talk about a double membrane, obviously outer membrane and inner membrane, the space between the two membrane is called as periplastid space. We have discussed about mitochondria also, and when we discuss about mitochondria, we know that it's also a double membrane structure. At tatu ki thake duta membrane o majot ekon space thake, and that space was called as what? PMS, right? So that was PMS, and this is periplastid space. Next, since it's a chloroplast, we know photosynthesis would be conducted here. So what is the product of photosynthesis? Obviously starch or the food or the glucose, and so you'll find certain granules that contain starch also, right? Next, in the chloroplast, the inside portion or the inner portion, we have a term for that, and that is what is called as stroma. So what is stroma? The gel-like substance or the inner content of our chloroplast is called as stroma. In the stroma, we see certain sac-like structures. Sac-like structures. A sac-like structures they have a particular term that is called as thylakoid. Inside it, what we see, the thylakoids could be arranged single, or they could be arranged in the form of a stack. What is a stack? One above the other, that is what is called as a stack. If thylakoids are arranged in the form of a stack, we don't call them as thylakoid, instead we use a word called as grana. So what is grana? A stack of thylakoid is called as grana. So this is a description. As far as the genetic material is concerned, it has circular DNA, and whenever we hear about circular DNA, we remember about that bacterial cell also. Right? We have talked about mitochondria. Mitochondria to circular DNA ase, chloroplast to circular DNA ase. And in case of a prokaryotic cell also, we have seen the presence of circular DNA. This is one point that is common, and that is the reason, if you remember, we have used a word called as endosymbiont of bacteria. Last class or a word use korisil mon dese when we were talking about mitochondria. So similarly, semi-autonomous cell organelle and endosymbiont of bacteria, these two sentences or these two words we have used during mitochondria class. Similarly, we are going to use exactly the same sentence in case of chloroplast also. What is the reason behind? The same explanation that we have given in case of mitochondria. Is the point clear? We'll be writing about it. So circular DNA thake, genetic material obviously in the form of DNA and also in the form of RNA. Talking about ribosomes, ribosomes present or absent, ribosomes present, it will be 70S or it will be 80S type. It will be obviously 70S type, because we know in a eukaryotic cell the type of ribosomes is always what, 80S, but inside the cell organelle ki thakibo 70S thakibo, kot thakibo, mitochondria thakibo aru chloroplast thakibo, right? Yeah. Once this is over, so what we will do is we will write the notes now. As far as explanation is concerned, this was the explanation about the structure. Another point from photosynthesis point of view, since chloroplast is the organelle where the photosynthesis process will occur, we know the entire process of photosynthesis has been divided into two parts, that is the light reaction and the dark reaction, right? Plant adale food bonabo lagile, we know it should have certain types of requirements, certain types of raw materials it is used, and tar pisore tar pora ki hoy food to bonabo pare. Similarly, ami jeneke bhat bonabo lagile we should have the rice, right? Ami saul kin to kini anibo laibo, we have to cook it down and then only will have the food, right? Similar is the case in case of plants also. They have certain type of raw materials, they will use those raw materials and ultimately they will prepare the food in the form of starch, glucose or whatever you say, right? As far as photosynthesis is concerned, broadly divided into two parts, that is the light reaction and the dark reaction, right? And we know food is obviously produced during the dark reaction and not in the light reaction. Because another duta part as a reaction to, kintu exactly starch ba glucose code produce correct, dark reaction not produce correct. So what occurs during the light reaction? Light reaction, a raw materials like use correct. For example, they will take in the light, because light obviously it comes from like the sun, or sun hobo lagile light to hobo laibo, day hobo laibo, so it's a part of a light reaction, right? So yate key question ahe. So if you talk about photosynthesis, it occurs inside the chloroplast. If you talk about photosynthesis, there are two parts. One is light reaction, the other is dark reaction. The question that is asked is, where does light reaction occur? Where does dark reaction occur? Photosynthesis kot hoy chloroplast. So light reaction, dark reaction kot hobo chloroplast hobo, but you have to be precise as to in which part the light reaction occurs and in which part the dark reaction occurs. As of now, what you do is stroma side or likhi thoi diasun. Site of dark reaction. Our thylakoid or lika site of light reaction. Right? So light reaction kot hoy chloroplast bhitor, dark reaction kot hoy taku chloroplast bhitor, but you have specific areas, right? If I ask the question, why does light reaction occur in the thylakoid? I think we know the answer. Kiho karne? Light reaction thylakoid kiyo hoy? Light reaction mane ki? Someone will have to take in the light, right? Light in the form of photons, right? So light to lobo lagibo, kune lobo light. Plants say light loy. This is something that we study. Plants absorb light and they conduct photosynthesis. What we understand by the line plants absorb light, kene ke loy tahate? They don't have a hand, okay, they will absorb because they will have the pigments, right? So, etia kotha to hol, light reaction if it occurs in the thylakoid, then there should be certain structures which will take in the light, and what structure we are talking about is only the pigments. So, where are the pigments present? Pigments are present in the thylakoid, and that is the reason why light reaction occurs in the thylakoid. They are absent in the stroma, so light reaction does not occur in the stroma. Clear? Next, again talking about a thylakoid, we will be writing it down later. When we talk about a thylakoid, the thylakoid is again a double membrane cell organelle. Chloroplast or bhitorot thylakoid thakibo. Thylakoid to kintu double membrane hoy. So when we talk about such a structure, we have something called as a matrix. Matrix ami buji pao in the inside content, and these two erutak ami ki boli kom? Membrane. So again the next point is, if I say the pigments are present in the thylakoid, you have to be precise. Thylakoid or kot present, bhitorot present, ne membrane or present. So always remember the pigments are present in the outer membrane of the thylakoid, right? So ebilak jagathe ki thakibo pigments thakibo. This is the part that we are going to do in photosynthesis again. As of now, since we are talking about this structure, we have to know all this. So ekhini loke light reaction hol dark reaction. Now, if I ask you, what is the type of reaction that occurs in Grana? Ki koba? Light reaction, because it's a collection of thylakoid itself. Thylakoid ki thakibo pigments thakibo. So jodi pigments thake tat ki hobo? Light reaction e hobo. So not necessarily exactly A2 question by A2 question ahibo pare. The question could be asked about Grana also. Right? It's obvious that obviously yate ki hobo? Light reaction. Now we have something called as certain type of channels or stroma lamellae, connection between two grana. Not necessary in a chloroplast okol duta grana thakibo. There might be so many granas, but in the diagram I am just showing 2 of them, right? So bahut grana thakibo, yato grana thakibo pare, yato grana thakibo, gutei grana bilakor majot ki hobo connection thakibo. Clear? So question keneke ahe from MCQ point of view, diagrammatic questions ahibo pare, A B C D D dibo, you have to identify the part. Second question could be what is the site of light reaction, what is the site of dark reaction. Third question could be single membrane or double membrane structure. Fourth question, type of ribosomes that is present, right? Fifth question, type of genetic material that is present. Clear? Okay. So this was the explanation. We'll write the notes. Are you new to the class? You came before? Online or offline? Both? How come I did not see you? With you? To me, ahisa. Board khon musibole jodi iman somoy lage keneke hobo paru kosun. Or maybe I should have some extra food, get some extra energy to rub the board. Certain types of markers, no, they are there. Sasha, it's not going. I think the ink is bad. Likha, the term chloroplast was given by online students. Do you have any doubts regarding the explanation or it's understood? Okay. Okay, very good. Okay, please write now. Okay, talking about shapes of chloroplasts, this is a part that we have also discussed during LG, right? When we have talked about LG, tate ami koisu. So since we have already discussed in the class, do you remember what is the shape of chloroplast that is found in case of higher plants? Higher plants also. That is the same type of chloroplast that is found in case of green algae also, because we know green algae has a relation with the higher plants. Evolve ki hoise green algae purai hoise, because if we talk about algae, the most primitive ki, we have 3 types of algae, no? Advanced kuntu, green algae. So if it is advanced, then it will have certain types of similarities with the advanced plants also. So one point of similarity is the type of chloroplast or the shape of chloroplast. Ki asile discoid. Discoid or discate. We have drawn this chloroplast, right? Ematro juntu akisu, it looks like a disc, yes or no? So such a type of a chloroplast is called as what? Discoid, right? Chlamydomonasত কেনেকুৱা আছিলে cup shell very good Please mute yourself. What is a stellate type of chloroplast? A star-shaped chloroplast is called as stellate. Rest of the information in the LG notes. Please refer to that. Remember the word mesophyll cells. Last class we discussed mesophyll cells. Ki asile? What is mesophyll cell? Jana ne kobo nwaro. Jedi kobo nwara it's okay. Janu ne na janu. Kod pai? Leaves or pai. The ground tissue in case of a leaf is called as mesophyll cells. Right? As far as the number of chloroplasts is concerned, iman specifically teneke question nahe, thik ase. But important, obviously, chlamydomonas, we always talk about the presence of one chloroplast, thik ase. Teneke spirogyra. Remember, we have drawn the diagram in spirogyra when we were talking about algae. What we said, we have drawn something like this, right? So tate ki thakibo? Eneke chloroplast thakibo, spiral, right? Ar tate apart from the chloroplast, you also see what? Pyrenoids, how many pyrenoids? Many pyrenoids. In case of chlamydomonas, how many pyrenoids? One pyrenoid. And from the plant kingdom portion, we have talked about anthoceros. Anthoceros is a type of a bryophyte, and what we have talked about is bryophyte has a algal character. What is the algal character of anthoceros? Presence of one chloroplast and one pyrenoid. Forgot. Anyway. Okay, so another point while we were discussing about spirogyra in the algae class, I don't know whether you remember or not. Obviously, chloroplast kenua thakibo, spiral type of chloroplast thakibo. As far as spiraling is concerned, whether it is left-handed spiral or right-handed spiral, hetu ami discuss korisilo. Left-handed. Okay. And mesophyll cells at kiman chloroplast ase, generally it's not asked. Okay? So you see this one and this one. Next, we'll be writing certain terms. Terms boli likha so, likhi pala first term likha, pheoplast, P-H-E-O, pheoplast. Okay, heard about the word called as pheophyce? Ki hoy? Fungi hoy? Brown algae, right? Pheophyce, rhodophyce, confused ho goisa no? Okay, so jedi moi pheo word to use korso, then we are talking about brown algae only. So the plastid of a brown algae is called as pheo. Okay? sorry. So, Pheoplast and Rhodoplast are the two terms that we have understood. We have a new term that is called as Etioplast. Etioplast logot we have two another terms that comes together, etiolation and etiolated plant. A etiolated plant have a etioplast. The plastid of a etiolated plant is called as etioplast, and the whole process is called as etiolation. What is etiolation? It is a process. Right? This process occurs in a specific type of plants that is called as a etiolated plant. Etiolated plant or ki thake? Etioplast thake. What is etioplast? Plast is a plastid. Etioplast mane ki? A chloroplast of deprived sunlight. Means what? If suppose okay, we will understand with the help of an example. Suppose we take two seeds, okay, starting from germinate, I mean duta seed loiso, and we allow both the seeds to germinate. One seed we place them in direct sunlight and the other seed we place them in a shade condition. Duta seed loiso, duitai germinate koribo, seedor pra to obviously baby plant develop koribo. It does not matter whether you put it under the sun or you do not put it under the sun, right? So duta loiso. So we have a seed here, we have a seed here. Etu ki hoise sun buli noko aru. Light or torot thoiso, etu dhora shade or thoiso, mane without light condition. So from here also a baby plant will develop. From here also a baby plant will develop. Will both the plants be similar? Ne okoman kiba difference thakibo. Difference thakibo. What is the difference? Okay, talking about the growth, the height, the thickness, hetu nohoy. Since we are talking about chloroplast, we are concerned with plastid, we are talking about the color. Let's answer it in the point of color. Duita plant or junto baby plant diate pora develop koribo, color wise different hobo ne nohoy. Hobo? What will be the color? Yet obviously it will be green because it has fallen sun, because it is under sunlight. As far as this is concerned, it will green hobo jano. Green nohoy. Etu ki hobo? Almost white or off-white or pale color, right? So this condition is called as etiolation, right? The starting from, if you put a angiospermic seed and allow it to germinate, starting from germination, if you put it under shade condition, the phenomenon or the observe the morphological features, the pale color that you are going to see, that is called as etiolation. Why it became pale? Because it did not receive sunlight, and you have to know that chlorophyll form koribo lagile. Chlorophyll korpra form kore. Chlorophyll obviously kiba ta raw material thakibo laibo na. You should have a raw material, and from there only the chlorophyll would be formed. So we have something called as a protochlorophyll. That is the compound from protochlorophyll ki form kore, chlorophyll form kore. This is a part again in photosynthesis that we are going to discuss. Chlorophyll corpora form kore, chlorophyll protochlorophyll form kore. Protochlorophyll will form chlorophyll only in presence of light. If protochlorophyll does not receive light, chlorophyll form no kore. Right? So edal plant to protochlorophyll ase, yatu protochlorophyll ase, yate green color kyo hoy gol, because that protochlorophyll was exposed to light. Right? So it became green, but in this case, ki hol, the protochlorophyll was there, but it did not get light, it developed a pale color, and these symptoms that is seen in such a type of a plant, the process is called as etiolation, the plant is called as a etiolated plant. Tamane ki hoy, plastut ki na thake, sunlight na thake. Right? Sunlight lobo pora tahator capacity na thake. So just remember the words etiolation, etiolated plant, and a etioplast. What is a etioplast? Chloroplast of deprived sunlight, right? Adult plant deprived sunlight mane ki? Shade condition, no? Adult plant ase, shade condition ase, tar bhitor to chloroplast ase. Kintu chloroplast tut ki nai? Sunlight lobo pare nai. So that is what is called as etioplast. Clear? Okay. Next. We will write certain points about the inner membrane and the outer membrane. তোমালোকে পইণ্ট ৱাইজ লিখি যাবা। ইমান স্পেচিফিকলি কুৱেশ্যন অহা নাই, তথাপি we have to study. Selectively permeable, permeable, I think we know the words permeable, impermeable, selectively permeable and differentially permeable. Permeability tells us about what? The transport of substances across a membrane, right? So, এখন membrane is if it allows all the solid, all types of liquid, all type of gaseous particles in and out, that is what is called as a permeable. Right? If we use the word selectively permeable, then we are confined only to the movement of liquids, right? Selectively permeable. Next, semi-permeable যদি হয় তেতিয়াহলে only liquids. Selectively permeable, some. I will be selective in nature as to what I want, I will take in. That is what is called as selectively, right? That is also called as differentially. We will be talking about it later. Next, what is the important point from this is the word purines. Remember, we have talked about purines during a bacteria class also. Right? So, another similarity between a chloroplast and a bacteria. Ketaman similarity to already palu, no? Regarding the ribosomes, regarding the genetic material. The third similarity we are talking about is the presence of porine in both the cases. Porines ki asile? The protein pores, and through these pores, substances move from one place to another. Right? So jihetu photosynthesis লগত related আছে, there are certain words that we'll study during photosynthesis class. For example, we have a particular enzyme that is called as Rubisco. For those students who have studied photosynthesis or maybe have an idea about light reaction and dark reaction, dark reaction kunubai to porisa? So light reaction, dark reaction or kind, remember certain words called as C3, C4 cycle, right? C3 cycle, C4 cycle, eblak ki hoy, enzyme operated, right? And you have specific enzymes for C3, specific enzymes for C4. C3 enzyme ki asile? Rubisco. This is our photosynthesis class. As of now, Rubisco very, very important enzyme for photosynthesis. If Rubisco is absent, in case of C3 plants, start of photosynthesis is not, right? So, in what is the question that is asked from MCQ point of view is percentage of chloroplast enzyme, right? Rubisco forms dash percentage of the chloroplast enzyme. So, you have to remember the 17%. Second is, it is the highest percentage, right? Out of all the enzymes, maximum ki thake Rubisco thake. Kot thake inside the chloroplast, kio thake karon photosynthesis conduct koribo lagibo, right? Next likhiba something about the thylakoid. Likhasan thylakoid boli likhi pelai likhiba site for light reaction or stroma ta mein likha nai niki site for dark. I think it's already written in the diagram, right? So just remember thylakoid boli likhi pelai first point likha site for light reaction. A2 class of generally no, because we have not done photosynthesis, no, kisuman words okman notun hobo, right? Once we complete this and then we study about photosynthesis, at that time you will be able to relate both the words, right? As of now we have certain words called as quantosome, certain things like light reaction. You have to know what is light reaction also. Okol light reaction hobo boli kole to nohobo no. The entire process you need to know, right? So from this chapter, iman details nalage, photosynthesis ami details korim. As of now, whenever we talk about thylakoid, thylakoid ki hoy double membrane. First point. Second, ki ase It is the site of light reaction. Why it is the site of light reaction? Because pigments are present. Where are the pigments present? On the outer membrane, right? Next, since the pigments are present, again this is an explanation again from photosynthesis. We know pigments are important for photosynthesis. Pigments ki kore, they will absorb light. So if I say pigments are present in the thylakoid, the next question that follows is, are the pigments present single or they are found in a group? Pigments bilak thake thik ase. Pigments boli kole pigments bilak single ke thake ne group or thake. Before answering that question, another point that you have to know is pigments square. So what is the pigment that we are talking about? We are talking about photosynthetic pigments. And whenever we use the word photosynthetic pigments, it's not only chlorophyll that is a part of photosynthetic pigments, aru tu ase, carotenoids ase, carotenes ase, xanthophylls ase, right? Everything together are called as what? Photosynthetic pigments. Yate moi specifically to A2 mention kora nai je chlorophyll present. Moi ki koisu pigments present. I did not mention it like this, saying that it's only chlorophyll that is present, right? So what we need to know, no doubt we are talking about it as pigments. The next point is pigments are always present in a group. Single pigment na thake. Eto jodi thylakoid hoy no, yate dhora chlorophyll thakile, yate carotenoids thakile teneke na thake. They will always remain in a group. Eto eta group, like that, right? That group of pigments is called as what? Quantosome. Right? What is quantosome? Another word to use it. It is a photosynthetic unit. Quantosome or meaning to ki? Actually, ami ebla words photosynthesis korim. Quantosome ki hoy? Photosynthetic unit hoy. And whenever we use the word unit, what we or to put the question like this, ami unit word to ketia use koru? When we have to measure something, unit of length, unit of height or unit of etc. Unit ami ketia jeta ami measure koru, right? Yate ami ki measure koru? So there pigments keta ase. What is the number of pigments? So we are measuring something. So we have used the word called as unit, right? So we are talking about the number of pigments that is present. We know that the pigments are present in a group. That group is called as what? Photosynthetic unit, or it is also called as a quantosome. If I say pigments are present in a group, the next point that we have to know is what exactly is present in the group or who forms the group. Group tut kun thakibo? Chlorophyll thakibo aru carotenoids thakibo, right? Aru specifically jabo lagibo je keita chlorophyll thakibo aru keita carotenoids, 230 chlorophyll molecules and 50. Again, another specific point, if you talk about chlorophyll, it is not only chlorophyll A that is present, there are 10 types of chlorophyll. So, 50 230 chlorophyll ase, that does not mean I am talking only about chlorophyll A, right? Out of all the types of chlorophyll, chlorophyll A is considered as a universal chlorophyll. Photosynthesis hobo lagile, chlorophyll A thakiboi lagibo, and that is something that is present in all types of plants, right? Junbilake photosynthesize kore. Kintu apart from chlorophyll A, chlorophyll B2 ase, Cu ase, Du ase, U ase, right? We have around 10 types of chlorophyll. So, I said 230 chlorophyll molecules would be present. The next question again, out of all the chlorophyll molecules, which one is most important? Remember something called as a reaction center. June photosynthesis korise. Okay, let's not waste the time now. Okay, as of now we understood it is a unit, it's a collection of pigments that is called as quantosome. It consists of chlorophyll and carotenoids. 230 chlorophyll means all the various types of chlorophylls B, C, D, heblak sob thakibo. Kintu within this group it is only one chlorophyll A molecule that will be present. 230 bhitorot chlorophyll A keita matro eta. Bakipur beleg chlorophyll. Are sobotse important ki chlorophyll A. And if you remember, reaction center ki hoy, chlorophyll A forms the reaction center. Clear? And coming to this sentence also, this sentence will also be hard to understand since we have not done photosynthesis. Enzyme complexes, electron carriers, coupling factors, everything related with photosynthesis. Again, the same question. Zedi photosynthesis porisa, chemiosmotic hypothesis porisa niki. Chemiosmotic hypothesis zedi porisa, you will know about all this. Right? It talks about what? How is ATP formed inside the chloroplast? ATP could form kore, mitochondria bhitor form kore, so it's a part of respiration. But we know ATP is formed during photosynthesis also. Our photosynthesis ATP produce koribo lagile, electron, protons move koribo lagibo, right? So just leave this part as of now. Photosynthesis class ami details kori. There is no point of discussing photosynthesis in cell, right? Okay, we have used the word called as lumen, right? Yate ami matrix word to use korna, use ki koriso lumen. Please pay attention to that word, right? Today, dark reaction occurs in chloroplast. Ba aru jedi khude you can dark reaction no hoy, pigments in the thylakoid are present in membrane ne lumen. So you have to know by the word lumen we understand the inner contents of thylakoid. Next point. So this towards goes with chloroplast also. Exactly the same thing we have explained during mitochondria. So I am not writing the explanation again. Jikine mitochondria likhisilu, exactly the same in case of this also, right? So semi-autonomous because they are independent, but still on they are dependent. Dependent on what? Dependent on the cytoplasm, dependent on the nucleus. Next is endosymbiont of bacteria because it's thought that they have the origin from bacteria or they have evolved from bacteria. So I can explain nokoru aru. Okay, next points is one. তোমালোকে point wise লিখিছা নেকি নে এনে লিখি গৈ আছা doesn't matter point wise লিখিছা যদি what is the number আৰু ময়ে points লিখি দিও it hardly matters no point লিখিছা নে paragraph লিখিছা okay আকৌ photosynthesis আগতে photosynthesis পঢ়িব লাগিছিল আৰু তোমালোকে তো অলৰেডি পঢ়িছাই so you should be able to answer granule chloroplast and agranule chloroplast a chloroplast containing grana a chloroplast without a grana right what is a grana a stack of thylakoid is called as grana so inside the chloroplast obviously thylakoid would have to be present but not necessary grana থাকিব Grana ketia form koribo jeta stacks ot thakibo right so there are certain cells inside the plant body where the chloroplast is present but inside the chloroplast the grana is absent so the question is asked regarding that agranule chloroplast is it is found in a particular type of cell called as bundle sheath cell if you know about C4 cycle bundle sheath cells janibo lage right as of now khali point to mukhosto korim photosynthesis ot ami clear hom Another term C4 plants যেহেতু আমি পঢ়া নাই photosynthesis এটা কোন problem হ'ব right so explanation about C4 আকৌ যদি মই এতিয়া কৰো photosynthesis নিচিনা class হৈ যাব no point right so plants has been classified into certain categories depending on what is the type of dark reaction it shows right photosynthesis to plant ot hoboe kintu dark reaction light reaction or dark reaction photosynthesis duta part right light reaction ot ki kore light loy dark reaction ot ki kore food prepare kore no food keneke prepare korise different plants will prepare in a different method right? So similarly, some plants are categorized as C3, some categorized as C4, some categorized as CAM plants. So আমাৰ এনেকুৱা বেলেগ বেলেগ plants আছে. So যোনবিলাক C4 plants থাকে, সেই C4 plants বিলাকৰ photosynthesis দুটা cellত হয়, এটা হল mesophyll cell, এটা হল bundle shaped cell, right? Whereas C3 plants বিলাকৰ photosynthesis বা dark reaction এটা cellot হয়, it's only the mesophyll cell, right? There are certain differences between C3 and C4. As of now, what you remember, there is a category of plants called as C4. Example of a C4 could be sugarcane, wheat, etc. These are C4 plants. C4 plants are at a special cell থাকে, that is called as bundle sheath. Bundle sheath cellot chloroplast থাকে, hey chloroplastor bhitor grana na থাকে. So it is called as agranal chloroplast. Our question from MCQ point of view, just etu line opore question to ahibo. Obviously the process description to nahe no. So they will be asking you, agranal chloroplast is a characteristic of dash cells. It's a characteristic of bundle sheath cell. Okay. Second point. We have a term called as chromatophore. What is chromatophore? It is a structure that is equivalent to chloroplast in case of bacteria, because we know if you are talking about bacteria, certain bacteria are photosynthetic. So bacteria photosynthetic নহয়, কিছুমান bacteria photosynthetic. যদি photosynthetic হয়, তাহাঁতৰটো এটা area লাগিব না, য'ত photosynthesis হ'ব লাগিব. So what is the site of photosynthesis? In case of higher plants, the site of photosynthesis is always the chloroplast. In case of photosynthetic bacteria, the site of photosynthesis is chloroplast present or absent? Obviously absent, right? So photosynthesis ক'ত হ'ব? Light reaction ও থাকিব, dark reaction ও থাকিব, bacteria তো দুইটা হ'ব. So light reaction ক'ত হ'ব? Wherever the pigments are present, right? So in bacteria you have a organelle that is called as chromatophore. Chromatophore of ভিতৰত কি থাকে? Pigments থাকে আৰু সেইকাৰণে তাত কি হয়? Light reaction. So we can consider this to be equivalent to chloroplast in case of bacteria. When we talk about bacterial photosynthesis in our photosynthesis class, তেতিয়া আমি specifically mention কৰিম যে light reaction ক'ত হয়, dark reaction ক'ত হয়. Bacteria বেলেগ বেলেগ জেগাত হয়, right? Our plants are যেনেকৈ it's always within the chloroplast, but in case of bacteria we have two different areas where the light reaction and the dark reaction takes place. That is something regarding bacterial photosynthesis. পিছত পঢ়িম. As of now, just remember the term. Not very important from examination point of view because etu iman specific questions aha nai. So etia মই যিমানবোৰ points কৈছো no, that jot whichever point I have said that we will be talking about it in details during our photosynthesis class, what you do is side or note details in photosynthesis will likhi thua. So when we do photosynthesis, at least you can come back to your notes and see whether it's clear or not, else you will forget, right? So it jumbilak points at moi koisu photosynthesis of clear kori side later in photosynthesis kiba eta likhi thoi dia. And once we do photosynthesis, it's your duty to compare and see whether you have understood or not, right? Quantosome, C3, C4, Rubisco, ebilak sob bostu photosynthesis logot related. Talking about the origin of chloroplast, the same explanation that we have given in case of mitochondria, right? Mitochondria notun ke to form koribo nware, no? It is from the pre-existing mitochondria that new mitochondria is formed. Similarly, it is from the pre-existing chloroplast that new chloroplast gets formed. So we have used the word called as self-duplicating or self-replicating. Eketai bostu. Okay. Next is, what is a grana? A stack of thylakoid is called as grana. Chloroplast, inside the chloroplast, grana could be present, could be absent, absent in certain cases only. In commonly, grana is always present, right? Next, talking about grana, two words has been associated with it, oppressed grana or non-oppressed grana, right? Oppressed grana or non-oppressed grana mane ki hoy? What is this? A grana, right? This part and this part, jot niki pressure nai, tagami ki boli kom, non-oppressed part of the grana or non-oppressed grana. If you are talking about this layer or you are talking about this layer, this one has been stacked above the other, pressure ase no, so tagami ki boli kom, oppressed grana, right? Just the two terms used. लिखिले आरो कंफ्यूज हो बाइटा नेलिखो PS1 PS2 আকৌ कंफ्यूज আকৌ photosynthesis होबो लागे just two words pressed grana and oppressed grana photosynthesis আমি details কৰিম so this is all about chloroplast right Just see whether you have any doubts regarding this or it's understood. If it is understood, we'll move to the next cell organelle, that is ribosomes. Heading koriba ribosomes. Bacterial chloroplast nahi, because that is a membrane bound cell organelle, no? Membrane bound cell organelle is not present in case of a prokaryote, right? So membrane bound cell organelle nahi, kintu again that's a confusion. In general what we say membrane bound cell organelle absent, kintu tothapiyo to photosynthesis hoy, so photosynthesis hobo lagile kiba eta to structure lagibo jot niki pigments thake. If you are talking about it as a chromatophore is also a cell organelle, taru to eta membrane thakibo. Right, that is a little bit confusing, but from the general point of view, chloroplast absent because membrane bound cell organelle absent, right. Next is ribosomes. Ribosomes boli kole the first point, what is the function of a ribosome? So it is also called as protein factory. Right. Second point, membranous and non-membranous. Non-membranous. Third point, types. Two types, 70S, 80S. One of eukaryote, both in case of prokaryote. Sorry. One in case of prokaryote, both in case of eukaryote. Next, 70S or 80S bully koa loge loge, what we understand, this is nothing but the Sedberg's unit or something we'll be talking about. Next, the structure of a ribosome always consists of two parts. Duta unit log lagi pelai eta ribosome form kore. You have a bigger subunit, you have a smaller subunit. One is dome shaped, D-O-M-E. I think we understand what a dome is. Dome ki hip, right? Balir dome bully ko no. So what is a dome? It's nothing but a hip, right? One is dome shaped, and above the dome shape we have a cap like structure that forms the smaller unit. At a dome shape, at a cap shape, right? And ihote ki produce kore ribosomes. Talking about the presence of ribosomes inside a cell, either it could be found freely, right? Cell to bhitor freely pabo pari, or it could be found inside cell organelles also. Mitochondria bhitorot thake, chloroplastor bhitorot thake. That does not mean ribosomes are present only inside cell organelle. They are found in the cytoplasm also. And if you remember, we have talked about ER. Endoplasmic reticulum koisilo. RER is something that we have discussed. RER or SER or difference ki asile? The presence and absence of ribosomes, right? So if you are talking about a cell, this is the nucleus, A2 whole tumar ER or jodi eto RER tetal tat ki thakibo ribosomes thakibo. So ribosomes junbilak cell organelles or bahirot thake, this is also considered as a type of free ribosomes. Two types of ribosomes, inside the organelle or freely. So freely could be here or freely could be stuck with the membranes also or on the outer surface of the membrane. So nucleus jodi monot ase nuclear membrane khonu ki hoy? Two layered. So nuclear membrane or ki thake? Ribosomes thake. So A2 ribosomes ot ami organellar buli kom ne free buli kom? It's a free ribosome. Clear? And heard something called as a polyribosome. Bahut ribosomes ekelege jodi thake, right? Kintu okol ekeloge thake buli kole nohobo. If suppose this is a ribosome, this is a ribosome, this is a ribosome, right? They will remain attached in a chain, mRNA, hebor ami pisot kom. So a structure tok ami ki kom? Polyribosome. Ami likhim olop pisot, thik ase? So polyribosome does not mean it contains only many ribosomes. It contains many ribosomes attached to this one. Tetia hai polyribosome, right? True or false die? Polyribosome is a ribosome. Now what is polyribosome? Polyribosome contains many ribosomes only. False. Polyribosome contains many ribosomes together with that mRNA, right? A string dalu to lagibo no, jot lagi ase. Okay. So these are the points. Ami eta notes likho. Okay. Another point is, since we are talking about 70S and 80S, no types of ribosomes, a S to ki mean kore? It is either called a sedimentation coefficient or it is called a Sedberg's unit. Why is it called a Sedberg's unit? Because Sedberg was the one who has discovered it. Sedimentation unit buli kiho karone kore? Because if you want to study the ribosomes, what you have to do? You have to take out the ribosome from the cell. Chloroplast পঢ়িব বিচাৰিছা, what you will do? You will isolate the chloroplast. Mitochondria, you will isolate the mitochondria. What we need to remember is the cell organelles that have a membrane, either it's a single membrane or it's a double membrane, it's easy to be isolated. You can just take it out, right? But in case of ribosomes, tahator ki na thake, okay? I mean membrane na thake. So if you want to take it out, tumi ki enege uthai loi anibo pariba niki? Noara. So what you have to do? You have to go for sedimentation, right? So you will take the cell sap, or sorry, not the cell sap, you will take the cytoplasm. Cytoplasm ot jonkhini ribosomes thakibo. Hey ribosomes khini ki koribo? You will put in a centrifuge, right? Centrifuge is a machine that ki koribo ghuribo and the heavier particles will settle down, right? That is the reason why we use the word S. Jonto heavy hobo, heavy ribosomes, test tube eta thakibo, test tube to tolor pine ki thakibo, heavy ribosomes will settle down and lighter ribosomes would be on the above. So since we are studying the ribosomes or we are isolating the ribosomes due to or by using the process called as sedimentation, so we use the word called as sedimentation coefficient. Clear? So ribosomes are likha protein factory. non-membranous support question है, also called as third point. As far as who discovered ribosomes, duta scientists naam disu, right? But the question is asked about only this one, because generally Robinson and Bowen report question na yahan. So our ribosome is composed of ribonucleoprotein. What we understand by ribonucleoprotein? RNA aru proteins. Is this over? How good? It like? Remember the word cytosol. Jar jar monot ase. Cytosol ki asile? Cytoplasm, protoplast. Ekhon chart disilu no. So cytosol, that liquid content with that organelles ami separate kori dile. Okol liquid content to ami ki boli kom? Cytosol boli kom. So talking about ribosomes, two types to be precise, 70S or 80S, কিন্তু কেতিয়াবা আমি 55S ও দেখা পাওঁ, right? 55S only present or characteristic of mammalian ribosome. Iman important নহয়. As far as ribosomes are concerned, we know either if it's 70S or whether it's 80S, in both the cases they have two units. 70S or দুটা part থাকিব, 80S or দুটা part থাকিব, right? So if you are talking about a ribosome as 55S, then obviously it will also have two parts, a smaller part and a bigger part, right? So it will have 39S part and 28S parts. Iman specifically aha nai. Maximum jedi questions ahe, the question could be asked, 55S type of ribosome is found in mammalian ribosomes, or is a type of mammalian ribosome. কিন্তু iman important nohoy, rest of the points are important. Next likhiba structure of ribosomes. So we know that it's made up of two subunits, duita subunit log lagi thake, and that structure is called as what? Dimer. What is a dimer? A bigger unit, smaller unit. This is what is called as a dimer, right? Aru ei dutak log lagi thakibole kihor upor based? Mg, magnesium concentration. Jedi Mg concentration lagatke komi jay, then both will separate. এইটো এবাৰ কুৱেশ্যন আহিছিলে clear we know 70s and 80s next we'll draw a chart 70s আৰু 80s compare কৰিম so ইয়াৰ পিছত chart বনাই is this over hold So, এটা সাইডে 70s লিখা এটা সাইডে 80s লিখা Huh? Huh? Camera কি হইছে? Camera on করা। কোনটা আছে বলবো? Camera off হৈ গৈছে, board দেখা নাই? অকল শুনা পাই আছা ন? Okay, just wait 5 minutes. Diagram nalage iman confusing. Again, so from notes point of view, bahut khini likhiso kintu iman details ot nahe. Details ot ki ahibo pare? The smaller unit of this, pata nahi. The larger unit of this. Similarly, the smaller unit of this, the larger unit of this. Can you hear, students? But you cannot see. You can see now? Is the board clear? Okay, okay. Then please write it down. Okay, what I was saying is, from examination point of view, what is important is the points that we have ticked. The smaller unit of 70s, the smaller unit of 80s, larger unit of 70s, larger unit of 80s. And maximum, eta question ever rahisile. Normally, if you see in all the cases, because we know ribosomes are made up of what? RNA and proteins, right? And talking about RNA, no, we know that there are different types of RNA, mRNA ase, tRNA ase, rRNA ase, right? So this is a thing that you are going to study in your genetics class, the different types of RNA, what is the function of these different types of RNA, etc., etc. As of now, we know if you are talking about ribosome, it has to have RNA, it has to have protein. So RNA kiman thakibo, proteins kiman thakibo, and in only one particular case where you will see 3 different types of rRNA. Baki bilakot if you notice, no, so eta types ase, etut 18S ase, tate duta ase matro 23NS, kintu question to ebar etu upor ahisile. The 3 types of RNA is a characteristic of 70S ne 80S, is a characteristic of 80S, aru eta jodi precisely ahe which part of 80S. It is a larger unit of 80S. Details mukhosto koribo nalage, you don't have to remember. 5.8S and 5S. হৈছে লিখা? And suppose functions of ribosomes puli kurile, synthesize proteins. There is nothing to write about that, right? So synthesize proteins. আকৌ এইখিনিত এক্সপ্লেইন কৰিব নোৱাৰো, you'll be learning these words যেতিয়া proteins biomolecules আছে, যেতিয়া proteins পঢ়িবা তেতিয়া তোমাকে ক্লিয়াৰ হ'বা, right? Just from this point, say from this chapter point of view, what we know, ribosome synthesizes proteins. Ribosomes could be free ribosomes or it could be organelles ribosomes, right? In both the cases, ki produce koribo, proteins produce koribo. Some proteins will stay within the cell and some proteins will go out of the cell, right? Another word that is called as a chaperone. What is a chaperone? Ako yaru details poribo nwara etia. A protein that helps another protein. One example of such a protein is chaperone, right? What is the function of a chaperone? It helps in folding of a protein. Now, what we understand by folding of a protein, A2 chapter to obviously details or nalage, ami biomolecules are for him. What you remember is ribosomes bilake protein synthesize kore. Protein synthesize koribole tahatok ki lagibo, RNA lagibo, messenger RNA. So, DNA থাকিব, DNA কিছুমান ইনফৰমেশনস থাকিব, সেই ইনফৰমেশন বিলাক কোনে লৈ যাব, mRNA লৈ যাব, translation, transcription, these are the words that you will study later in your genetics class. As of now, যে each and every information এটা সেলত কি protein produce হ'ব, ultimately signal to কোনে দিয়ে, genes এইটো দিব, right? So genes বিলাকক কিছুমান codes থাকিব, এই codes বিলাক কোনে carry কৰিব, mRNA carry কৰিব, mRNA, then again we have certain process called transcription, translation, etc. Shortly এই মেসেজবোৰ কোনোবাই লৈ গৈ পেলাই আলটিমেটলি কি ফৰ্ম কৰে, যে তাহাতে proteins বিলাক ফৰ্ম কৰে। যেতিয়া first proteins বিলাক ফৰ্ম হয়, they are always formed in a linear way, right? এটা এনেকুৱা single line. কিন্তু পিছত কি হ'ব, প্ৰটিনৰ বডিটো লাগিব, ফাইনেল প্ৰটিনটো আমাৰ সদায় কি হয়, এটা এনেকা folded area, it's a unit, 3D structure হয়, right? Initial protein যোনটো ফৰ্ম কৰে, that is a linear structure. Final protein is a 3D structure, it's a folded protein. বহুত বস্তু লগ লাগি পেলাই fold হৈ পেলাই protein কৰিব। এই folding process of কোনে help কৰিব, chaperones হত। Another point is, if the proteins that are formed does not get folded, then what happens? Then there is a tendency that the proteins will convert itself to prions. Remember the word prions we have discussed during virus, right? Subviral particles of eta word ami ki porhisilo, prions. Prions ki asile, infective protein molecules, right? So inside a cell, prion kune form koribo, protein A to form koribo. So which protein? Junkhini protein were not folded, right? So jedi folding of a protein ba protein to proper structure develop nokore, tetiahole there might be chances that that protein would be dangerous to the cell, would act as a prion. Clear? Folding of proteins, biomolecular class, right? So this is all about ribosomes. So up till here what we have discussed is, we have talked about the boundary of a cell, we have talked about the cytoplasm, protoplasm, etc. and we have talked about the different organelles that is present inside the cell. Next part of the lesson we have to talk about something called as cytoskeleton, right? So I think we have time. So heading koriba cytoskeleton. বন্ধ কৰি থৈ দিলা নেকি? Yeah, please ask. Free ribosome synthesizes non-secretory proteins aru junbilak ribosomes organelles bilakot lagi thake ba humai thake tahate secretory proteins synthesize kore. What is secretory, what is non-secretory, biomolecular class of confirm hoba. As of now, not a very important point. Okay. When is your next class? 6:30. Enough time. Let's keep on writing and writing and writing, no? We have to finish the course, so like okuman to extra ke janibo lagibo, poribo lagibo, no? Okay. Etia cell kar hoise, class of cover hoise, cell school of karut mar hoise, cover hoise, and baki rest of the students, so you all know what a cell is. Okay. Whenever we talk about cytoskeleton, what we understand? is the framework of a cell. Human beings or jeneke skeleton ase, cell at proper shape and size thakibo lagile, they will also have to have a skeleton. The next point, if you are talking about a human skeleton, it is made up of what? It is made up of bones. If you are talking about a cytoskeleton, it is made up of what? It is made up of certain structures called as microtubules, microfilaments and intermediate fibers. Right? Tenukwa dhoroner kisuman structures thake, kisuman tube like structure thake, kisuman thread like structures thake, right? So these are categorized or named as microtubules, microfilaments and intermediate structure. Ultimately, ei tintai combine kori pelai ki form kore cell to cytoskeleton poribo. Next, what we need to study in this lesson is some points about the three types of structures, microtubules or microfilaments or hekini. Next is something called as the cytoskeleton of flagella and cilia and cytoskeleton of a basal body. If you have read this part before, have you come across a word called as 9+0 organization and 9+2 organization? Cartwheel shaped structure. Eblak words ami janu, right? So this is the part that we are going to discuss, right? When we talk about a basal body, first of what we understand by the word basal body is something that is related with cell division, right? Cell porha pisot ami ki porim, cell division porim, tat ami kisuman words porim, for example centriole, if you know the word centriole, right? Something that's related or something that's present near the nucleus. It helps in what? It helps in spindle formation during the process of cell division. So basal body ki hoy, first point. Second, basal body to dekhat keniko, how does it look like? It is made up of what? That is what is called a cytoskeleton. Similarly, we know what a cilia and a flagella is, the locomotory organs, right? So cilia and flagella bhitor structure to keniko. So this is something that we are going to study. One would be 9+0 and the other would be 9+2. So 9+0 puli junto structure thake, I think you all have seen something like this diagram. I am not drawing the entire thing, right? Eneke kiba eta cartwheel, no, ghura gari sokar nisnake eta bostu thake. So this is what we are going to study. So heading korason cytoskeleton. The cytoskeleton is made up of some proteinaceous filament cytoskeleton is found only in, ami cytoskeleton code dekha pam, eukaryotic cell, prokaryotic cell or dekha na pam. intermediate fibers, huh? কাম কৰা এই বস্তুটো নিলিখিবা এতিয়া এই দুটা পইণ্ট এইটো লিখিলা ন এইটো নিলিখিবাচোন এতিয়া বিকজ ডায়মিটাৰ দিয়া আছে ন ডায়মিটাৰ দিলে we have to be specific কোনটো proteinaceous filamentor আমি diameter লিখিছো because we know we have three types right microtubule আছে microfilament আছে আৰু intermediate fibers আছে so এতিয়া নিলিখো আমি next microtubules or point লিখিম right next heading koriba microtubules and as the name suggests tubules ase jehetu so it is a tube like structure and whenever we talk about a tube like structure bhitor to ki hobo it is hollow right when we talk about a filament the inside of a filament is not hollow it is solid right so if the proteinaceous filament is hollow that is called as a tubule if it is solid it is called as a filament or fiber microtubule heading kora লিখিলো মই বুজাই দিম protofilaments huh What is a microtubule? A microtubule is a proteinaceous structure. How does the structure look like? It is a hollow structure, so we understand it's tube-like, right? This single tube is made up of many subunits. Total keita subunit 13 ta horu 13 ta pipe logai pela eta bostu form kore, that forms a microtubule. These are called as protofilaments, and if you talk about protofilaments, protofilament also have certain type of subunits. So eta dekhisu kintu yate duta thakibo, right? Iman details jabo nalage, khali what is important is protofilament kiman ase hebilakoru questions nai ha. Question ahise the type of protein that is present in case of a microtubule. So it is tubulin protein. If you have noticed, no, yate ami ki likhisu joson, we are talking about microtubule. It occurs in cilia, flagella. We understand cilia, flagella, buji pao, centriole. I have already said nucleus osor thake, a characteristic that is present only in case of an animal cell, absent in case of a plant cell. Mitotic apparatus, we understand spindle fibers, right? Due to spindle fiber during the process of cell division, right? Have you heard something called as prophase, metaphase, anaphase, telophase, right? So what we understand, chromosomes pila ki hobo lagibo, separate hoi pila duta cellot jabo hoi lagibo, no? So who helps it? It is the spindle fiber, and the spindle fibers are called as mitotic apparatus. So those spindle fibers are made up of what? Those made up of microtubules, right? Next, basal body will explain when is a centriole called as a basal body. Basal body to ki hoy? Centriole to ke ami basal body boli kau, provided it has a different position. When it is near the nucleus, it is called as centriole. When it is at the periphery of a cell, it is called as basal body. We'll talk about it later, right? Next is swab. Now, talking about this, no, flagella is a locomotory organ. It is a characteristic of prokaryotes and eukaryotes. Both. You cannot be giving a single answer, no? A bacteria to asile, right? We have certain types of other organisms, algae to ase, right? Gametes bilakot ki thakibo, flagella thakibo kisman gametes. So flagella, one point of difference between a prokaryotic flagella and a eukaryotic flagella, the type of protein that is present. Tubulin protein or flagellin protein. So tubulin protein is a characteristic of ki eukaryote, right? Eukaryotic flagella ki thake, tubulin thake, prokaryotic flagella ki thakibo, flagellin protein, right? Hetu point hai besi important. Baki iman bor important nohoy. What is the time? 5:59, 1 minute, likhi long. Okay, no need to forget. I think that's all for today. Rest of the thing we are going to continue in the next class, right? So next class what ami ki korim, we'll be talking about the remaining part of this, and we'll draw the structures, that 9+0 and 9+2 organization, right? So any doubts? For the next class, functions of microtubules heading kore thoi dia, I'll forget. Functions of microtubule. Okay, thank you.